Now that we have the basics of DNA structure down, Chapter 10 delves into the issue of DNA replication. How do we make that new DNA that is a part of every mitotic or meiotic division? And the image here is, it looks kind of basic and simple, but what this is a strand of DNA that is being replicated such that after that split, you now have two identical copies of the double-stranded DNA molecule. And so we're going to dig into exactly how this happens. So how is DNA replicated? Well, before we dig into the real fine scale, the nitty-gritty of DNA replication, I want you to think about a double helix. Think about that forked image I just showed you on the previous slide, and how would you replicate it? And the interesting thing is that there are multiple ways that this could be achieved. And researchers were faced with differentiating between these options and identifying exactly which of them truly takes place within a cell. So if you see here, we have three different double-stranded molecules, each of them indicated by a pink double helix. And this figure illustrates replication of DNA going from one double-stranded molecule to two double-stranded molecules in three different ways. And they're classified as being either conservative, semi-conservative, or dispersive replication. And so if you think about this double-stranded DNA molecule, the goal is for there to be two of them after DNA is replicated. Okay? And now what I want to be clear about is when you're looking at this double-stranded DNA, you need to understand that both of these strands came from one parent. They represent one chromosome. All right? It's not that each of your parents gave you one strand of a chromosome and they bind together to form a single double-stranded molecule. They each gave you a double-stranded copy of each chromosome. Okay? And so what we're looking at here is just, say, considering the copy of chromosome number 1 that your mother gave you. It's double-stranded, and we need to make a copy of it so that the two cells that result from the mitotic division that's coming each end up with a copy of that chromosome that is identical to the one your mother gave you. And so if we look at the left version of replication, this is one of the ways we could go from one double-stranded molecule to two. And this would be copying the original double-stranded molecule and creating a new molecule, both strands of which are new. So the new here is indicated by blue. So you can see you have the original pink one and a new double-stranded copy in which both of those strands are new. That is conservative replication. Semi-conservative replication, indicated in the middle here, is when you go from having a single double-stranded molecule. That molecule splits apart into two single strands, and a new strand is replicated that is complementary to each of them. And so each of the two resulting double-stranded molecules has one new strand and one original strand. And then the third, on the far right, dispersive replication, is kind of a mix of both. You'll see that each of those double-stranded molecules is part pink and part blue, but you'll notice that each section, both strands are blue or pink. What has happened is little chunks of it are original on each double-stranded chromosome, and some segments are double-stranded new DNA. So the research that took place in order to determine which of these is the mechanism utilized by living organisms to replicate their DNA is what we're going to cover here in a moment. But I'm going to answer the question first and tell you that DNA replication is semi-conservative. That middle option, that middle pattern, is what we see in the real world. And I'll show you how that works here. And the way it works is a double-stranded molecule is split open, So those hydrogen bonds between the G's and C's, the A's and the T's, are broken. Those pink sugar phosphate backbones of each strand are pulled apart from one another, and so we now have these exposed single strands of original pink DNA here, and new complementary blue strands are created. You can see that if you look at the bottom pink strand, that first nucleotide on that bottom pink strand was an A, okay, in the bottom far right. If you look up at the top right pink original strand, that first nucleotide was a T. That makes sense, right? The A and the T would have bound together. They are complementary to one another. But you will notice now that a new strand has been synthesized that is complementary to these. So the top blue strand starts with an A that is paired up with that T. The bottom blue strand starts with a T that is complementary to the original A. So if you look at this and think about it for a bit, the top blue strand is identical to the bottom pink one. And the bottom blue strand is identical to the top pink one. Okay? And this is how semi-conservative replication works. Now, we're going to get into this in a great bit more detail, but first I'm going to tell you how scientists determined that this was indeed the way in which DNA is replicated. And they did it, these two folks, Meselson and Stahl, ran an experiment, again with bacteria, in which they included in the media an isotope of the element nitrogen, a heavy isotope, okay? So the isotope N15 is heavy, and so they grew up a bunch of bacteria, and then they took some of those bacteria and put them into a culture in which the only nitrogen available is the N14 isotope, okay? So it's a lighter version of nitrogen. And if you'll go back and you'll remember the structure of DNA, there are lots of nitrogens in each nucleotide. And so when these cells are replicated and they're making new DNA, when they make that new DNA, they have to take it, these raw materials, this lighter nitrogen, in order to make that new DNA, okay? And so what you're seeing below these flasks, where the cells are growing in a medium that originally had heavy nitrogen, now has a lighter version of nitrogen. Below that is a centrifuge tube. So you might have used a centrifuge in the past. It's basically a benchtop machine in which you can stick a tube, and it has a rotor in it that spins at exceptionally high speeds. So think of it kind of as a horizontal tire, if you will. So it spins around really fast, and what you do is you stick tubes in it, kind of facing laterally, facing to the side, okay? And you spin that centrifuge super fast. And what it does is it pushes heavy things to the bottom of that tube. And in these tubes, they put some of the DNA that they had extracted from these bacteria in each of these flasks, from generation 0, generation 1, 2, and 3. And they put that DNA in these flat vials that are going in the centrifuge, and they spin them. But what's in that tube is kind of a thick matrix, kind of think like a sponge or a jelly. And so not everything that you put in there is going to spin through to the bottom. It has to be kind of heavy to push all the way through to the bottom. The lighter it is, the less it's going to move from the top of the tube to the bottom. And so you can see the gravitational force direction indicated by that arrow. So as they spun this DNA in these tubes, the heavier the DNA was, the further it would move through the tube. And they could look at these tubes after spinning them and see how far the DNA pushed down through this gel matrix that's in the tube. And after generation 0, that DNA was heavy. It was all made with nitrogen 15. And so it pushed all the way down into that tube. But after generation 1, The only observable band of DNA after they spun it down, none of it went all the way down, none of it stayed at the top. It kind of went halfway down the tube, meaning all the DNA in there was of some intermediate weight. Okay? Now, this is going to make sense in a second here. All the DNA was medium weight. None of it had that original heavy weight. Look back at the conservative replication example, whether you're looking in the text or on the slides here. Those instances of conservative replication, you're still going to have one whole heavy double-stranded molecule and one light one. That's not what they observed. And then after generation 2, they observed again this medium weight band, and then they observed a new very light band of DNA. Okay? And in generation 3, they end up with more of the light stuff and proportionately less of the intermediate weight stuff. So what's going on here? What were they able to infer from this? Well, look at what's going on with the DNA. You start with, in generation 0, heavy DNA. Every strand is heavy and pink. But after one generation of semi-conservative replication, you end up with all the DNA being comprised of one pink original heavy strand and one new light blue strand. Okay? So all the DNA is half pink, half blue. Okay? All the DNA is of intermediate weight. And so when they spun it down, there was only one band in the tube, and everything had that same intermediate weight. But take those two double-stranded molecules from generation 1, and let's go through replication again. Replicate each of those. Because when you think about what those represent the DNA in 2 different cells. And going from generation 1 to generation 2, each of those cells is going to replicate their DNA and become 2 cells. So look what happens when you go to the right. What has happened is you took, let's just consider that very top double-stranded, half pink, half blue molecule, split it apart, and add a new strand to each that is complementary. What you got is a pink strand to which you are adding a complementary pink strand, also light. I'm sorry, a blue strand to which you are adding a blue strand, so they're both light. You can see that's the top double-stranded molecule. The other strand is pink, and to it you're adding a new blue light strand. So this top double-stranded molecule will yield, from generation 1, will yield those top 2 double-stranded molecules in generation 2, one of which is 100% all new, all light DNA, okay? And the other is half original generation 0 pink heavy DNA and half new lighter DNA, okay? So this experiment showed them that semi-conservative replication is what's really going on. At the same time, Taylor, Woods, and Hughes ran a complementary experiment in plants to make sure the same thing is going on in eukaryotes as is going on in prokaryotes, like the bacteria that Messelson and Stahl used. And so what they did was they radioactively labeled hydrogen in these plants and then allowed them to replicate their DNA. And so if replication was conservative, what you would see is something like on the left, okay? You would see the one chromosome being labeled radioactively and the other not, okay? But what they actually saw is something more like on the right, where there is radioactive labeling on both chromosomes, on all the chromosomes, okay? And this second part down here shows how there was actually crossing over in these chromosomes. I think the clearest example is probably the Messelson-Stahl experiment, but go ahead and read about the Taylor-Woods-Hughes as well. Okay, so I zoom in here on the image that they provide of these chromosomes with these radioactively labeled bits, and you can see the distribution of this radioactivity on the chromosomes. So what we have here is an illustration of how circular chromosomes in prokaryotes are replicated. And it's slightly different than in humans because ours are long and linear. Circular chromosomes have both an origin and a termination site where replication begins and ends. And so replication starts at the origin and works its way around the circle in both directions, splitting apart the original pink DNA strands and creating new blue ones, going in this instance to the right and the left. The spot where the DNA has been split is called the replication fork. So in this, you can see there are two replication forks in these circular chromosomes. And as these replication forks move around the plasmid, okay, we end up with two, as soon as they reach the termination site, we will end up with two double-stranded circular chromosomes, each of which contains one original pink strand and one new blue strand. Okay. Now that's DNA replication at a high level. What we're going to talk about now is how DNA replication gets started and actually works at the level of a single nucleotide. Okay, so we're going to zoom in to the DNA strands, and we're going to consider the first observations and conclusions that were made based on experiments with prokaryotic DNA, so bacterial DNA, and the observed effects and input of DNA polymerases, an important enzyme that we're going to talk about quite a bit. So here's what happens. Let's just consider only one of those original pink strands. What we need to do is make a double-stranded copy of DNA using only that one original strand. Because remember, what happens is that replication fork forms, these double-stranded pink molecules get ripped apart, all those hydrogen bonds are broken, but all of those nucleotides are hanging out there. And what they do is they provide what is called a template. You might think of it as a blueprint for how should the cell create this new strand? What should the nucleotides be in this new blue strand? And the cell doesn't have to, quote unquote, remember what should be there or what should be lined up complementary to this original pink strand. It is able to read that pink strand and bring in nucleotides, so G's, A's, T's, and C's, that are complementary to what's there on the pink strand. Okay, so that's how this is going to work. But what we are doing now, before we think about the G's, A's, T's, and C's themselves, are how does this blue strand actually get larger? How do we go from it being 7 nucleotides, as it is right here, to being 8 nucleotides long, and working its way all the way down and making a long, complete, complementary blue strand? Well, it does it by taking this original pink strand, what we call the template, or template, and you need the template, and you also need what are called DNTPs, dideoxyribonucleotide triphosphates, okay? So these deoxyribonucleotide triphosphates are present in the cells. So you can see there's DATP, DTTP, DCTP, and DGTP, all of those in parentheses in the middle there. Basically, these are all the G's, A's, T's, and C's. And the reason it's TP, triphosphate, remember when we learned about the structure of nucleotides, and I told you we will talk about them in either a form with 3 phosphate groups or just 1? They all start out with 3 as triphosphates, okay? And when you have these deoxyribonucleoside triphosphates, these DNTPs, and a template, when you throw DNA polymerase into the mix, you can glue them on to the end of this growing new blue strand of DNA, okay? And look at what that reaction looks like. When you have those 3 things, the new nucleotide gets incorporated, it gets attached to the blue strand, and look at the byproduct to the far right. You have 2 phosphate groups that have been released. Remember, this is what powers this reaction that gets these glued into position, okay? So nucleotides are added one at a time, and as the cell figures out what needs to be added, whether it's an A or a T or a C or a G, there is the enzyme, the DNA polymerase, that's doing this work, that is attaching this nucleotide. It is also determining what's across the street, basically, what's over on the pink original strand. And if it's an A, then the polymerase will bring in a T, which is complementary, and glue it on there. So let's look at a single strand of DNA at the finest level. You can see here it consists of phosphate, sugar, right? And off of each of those sugars on the 1' carbon is that base. Now, if we are to take these 2 nucleotides, this A and T, and we are to add to it this C, a C is the next thing in line, okay? You could look at this and you know that they connect to one another on the 5' and the 3' ends. But do you know right now whether this new C nucleotide is going to be added up to the top above that A or to the bottom below that T? Because it can't go both ways. There's only one way, one direction that DNA can be made. DNA is made in the 5' to 3' direction, from the 5 to the 3. And what that means is you add new stuff to the 3' end, okay? What was added is this DCTP. It was added to the 3' end of the T, okay? That was at the bottom of that 2 nucleotide strand. You can't add it up to the 5' end. That's not the direction that DNA or RNA is made, okay? So it is really, really critical that you understand the orientation of DNA and the direction in which it grows, okay? From the 5 to the 3. And I need you to understand that the new stuff is added to the 3' end, 3' end, okay? So we will dig into that a bit more as we get further along. You'll see why it's important. But I'm going to come back to this enzyme that I talked about, polymerase. And within the bacteria that were initially studied when the replication of DNA was explored, these bacteria have a number of different polymerase types. They have DNA polymerase 1, polymerase 2, and polymerase 3. And the abundance of each of those and their activities are quite different. So what you see here is a table that indicates the activity of each of these molecules, these different types of DNA polymerase. And what I really want you to pay attention to and understand, or be aware of at least, is not the fact that they can polymerize in the 5 to 3 direction. That's what that second line indicates. So that means they can add a nucleotide to the 3' end of a strand of nucleic acid. The exonuclease activity is something that I want you to familiarize yourself with. What this means is that it means that as they're making new strands of DNA, if mistakes are made, they can fix them. These polymerases recognize and are able to repair, so they remove a mistake. If they put a C next to a C, if they paired up a C and a C together, they can go back and rip out that C that they put in and put in the proper G that should be there. The two different lines here indicate whether they can do it only in the 3 to 5 direction, so reading backwards, or can they do it in the 5 to 3 direction as well. So just understand that there are multiple polymerases and they differ in the way that they work or the things that they are capable of, but I want you to understand that polymerization always takes place 5 to 3, but that exonuclease activity can work in both directions. Okay, so now we're going to talk about replication itself and some of the issues that the cell faces given these limitations. And the primary limitation I'm talking about is the 5 to 3' polarity restriction in the direction that DNA is synthesized or made. So let's talk about all of the things that have to be dealt with in order for a double-stranded DNA molecule to be semi-conservatively replicated. And you're going to see this slide multiple times. I'm going to work through each of these bullet points, one or a few at a time. Okay. So the first thing we have to deal with is that double helix, that original pink double-stranded double helix, has to be unwound. We have to get it apart. We have to break a boatload of hydrogen bonds, right? And then it has to stay apart. Those hydrogen bonds are kind of sticky, so if you just break it apart and leave it, it's going to come back together. So you got to break it apart, keep it apart, and we have to deal with supercoiling downstrand. Now, what does that mean? It's a difficult concept to explain to your generation because the best example I have is the old telephone cords. So when a phone had a cord, good Lord, I can't speak. Those cords were coiled, okay? And they would frequently get tangled up, okay? And if you simply grab that phone and just pull it away from the base of the phone, thinking that just yanking on it is going to uncoil it, you're just going to make it worse. You're going to tangle it up. The same happens with the double-stranded DNA molecule. If you just grab the two ends of it and pull them apart, what it does is it pushes the knot, the coiling, tightens it up downstream. You've got to deal with that. Okay, so we're going to talk about those two first. And those two issues are dealt with by these three things illustrated here. Okay, we're going to start with the two that are on the left. There's an enzyme indicated here, helicase. So what helicase does is it opens up the double helix. It breaks those hydrogen bonds. That's all it does. And you can see it here at the replication fork on the far left. And imagine it just simply plowing through from your left to your right, breaking every hydrogen bond in its way and pulling apart these strands, keeping them single-stranded. Now, behind it, or just above and below it in this image, you will see single-stranded binding proteins. These proteins do just what it says. They bind to single-stranded DNA and they keep it single-stranded. They hold it apart. They keep it away from the other complementary strand that's below or above there and make sure that the hydrogen bonds don't reform, undoing the work of the helicase before the DNA can be read and a new complementary strand created. Okay, now the final bit or piece of this puzzle illustrated here is the role played by DNA gyrase. And now look at the far left where the DNA has been unwound. If you simply took each of those ends and just yanked them up and down without untwisting, you're going to end up with just compressing all the twisting to your right until it just becomes a tangled mess. And so what DNA gyrase does effectively, as this tension builds up in the double helix because the helicase is pushing it apart, it clips the double-stranded DNA, lets that supercoiling kind of spin itself out, and glues it back together. Okay? So it allows the DNA to unwind by the action of the helicase without getting all tangled downstream. Okay? All right. So the next thing we have to talk about is the need for a primer. Okay? So the issue we face here, and one that I haven't touched upon yet, is that when you are adding DNTPs to make a DNA strand longer, you have to add it. You can't just bring a T in and attach it to an A on the template strand. You have to also attach that T to the 3' end of the base just upstream, the previous one. Okay? So just like that first image where I showed you the work of DNA polymerase I and it added that new green nucleotide to the end of the growing blue strand. That's great. That works perfect because that green nucleotide had a complement over on the pink strand and there was a 3' end that it could be glued onto on the new blue strand. Okay? But what about here at the beginning, at the far left? Okay, so what we're looking at here, let's forget the coiling and the twisting and the other strand. We're just looking at one template strand of pink DNA here. And we are making this new blue strand. Okay, now you will see that only the right 2/3 of this new strand are blue. The initial bit to the far left are an orange in color because they're not DNA. They are made of RNA. And you will remember the only difference between DNA and RNA is what's hanging off that 2' carbon and whether they have thymine or uracil. Okay, so it's very similar. It does much the same thing. But guess what? We were able to start making this new strand out of RNA because that very first nucleotide that was stuck in there, it doesn't care if there is a 3' end hanging off to our left for it to be glued to. All it needs is a complementary nucleotide over on that pink template strand. Okay? So unlike DNA, RNA doesn't need that existing 3' end to be attached to. And so the way we get DNA synthesis started is with what is called a primer. And the primer is, in this case, made out of RNA. Okay? So you already know that DNA polymerase plays a role in DNA synthesis. It glues on chunks of DNA to existing ends. Okay? But in order to get this started, we need an RNA polymerase. Okay? That glues RNA together. And this particular one is called primase. So there are different kinds of RNA polymerase. Remember, DNA polymerase, there was type 1, type 2, type 3. RNA polymerases, there are a number of different kinds, and one of them is primase. It's used only for this purpose, and its purpose is to put a few nucleotides in here, start making a new strand. So it creates this orange strand with, let's say that's 10 nucleotides of RNA. And it gets a little ways along, and then DNA polymerase notices and says, hey, thanks for doing this. There's a 3' end hanging off of the last RNA nucleotide that you added. I can now add DNA nucleotides to this and create a long blue DNA strand that is complementary to the RNA. Okay? So this is an important restriction in the ability of DNA polymerase to synthesize DNA. They have to have a hanging off 3' end. Now, the other thing I want you to notice here is that this new strand is being synthesized from your left to your right. Remember, we talked about DNA is made in the 5' to 3' direction. All right? And so this strand has been synthesized, but look at the original template strand. Remember the orientation of DNA strands? They are anti-parallel. So that original strand from your left to right is 3 to 5. So if I just show you a template strand that looks like this, 3 on the left, 5 on the right, what direction is the DNA going to be made, the complementary strand? It's going to be made from your left to your right. People memorize it's made 5 to 3. But where they get mixed up is they look at that pink strand, the 5's on the right, the 3's on the left, so I'm going to make a new one from right to left. That's 5 to 3. It's not 5 to 3 of the thing you're copying, it's 5 to 3 of the thing you are making. Okay? And what is happening as this DNA is being made 5 to 3, realize that the polymerases are reading that pink template. That pink template is being read 3 to 5. It is being read from your left to your right. So DNA is made 5 to 3. It is read in the 3 to 5 direction. Both of those are your left to your right. Okay? So in all of these examples that you see and everything you look at online, please be sure that you understand and follow that 5 to 3 prime synthesis restriction and then reading 3 to 5. All right, so now we understand that synthesis has to be started through the use of a primer. Okay? We also now need to think about the fact that because of this polarity restriction, that it can only be made 5 to 3, and that the two template strands of this original double-stranded molecule are anti-parallel, that we can only have what we call continuous replication on one of the strands. The other cannot be continuously replicated. And let's look at why that is. Okay, so here's an illustration showing we split apart the DNA a little bit, and we have to make the new DNA in the 5 to 3 prime direction. So let's look at that top illustration. The bottom new strand is made left to right. The top new strand is made right to left. Right? They have to be made in opposite directions because those template strands run in opposite directions. So that's all well and good, but what happens when helicase pushes further downstream? Look at the middle image. Further downstream, that bottom new strand, the one we call the leading strand, just keeps chasing the helicase. It follows that split, and it is continuously synthesized forever and ever until it reaches the other end. However, the upper lagging strand, in replicating that, as more of the template pink opened up, we had to go in and, because of the direction restriction, that new shorter arrow to the upper right there, we had to start with a new primer and synthesize new DNA from right to left. And that new DNA was synthesized until the polymerase ran into the primer that's already there. Okay? And so now we've got two discontinuous fragments on that new strand, that new upper strand. And then go down to the bottom illustration, helicase pushes further to the right. We have to do it again, make a third discontinuous fragment. The bottom new strand is still continuously synthesized. And what we end up with at the end of this process are discontinuous fragments that have primers stuck at their beginnings, okay? And the other strand, the bottom strand that's been continuously synthesized, has only one primer at its very far left end. And this is something that we're going to talk about here a fair bit. So here's an updated illustration from your text illustrating this the action of DNA polymerase. They talk a good bit about this DNA pol-3 core structure and the holoenzyme. So all of these bits that are in there, the green, the orange circle, the pink three-fingered thing, the purple connectors, those are a bunch of different proteins that together form the mechanism that initiates this replication of both strands, the leading and the lagging strand, in the proper direction. Now you'll see that lagging strand has to be kind of looped so that it can be synthesized as the sliding clamp loader keeps pushing to your right. But both of these strands new strands are being synthesized in the 5 to 3 prime direction. Okay? All right, so now we've covered these first 4 points, and now we're going to talk about the issue associated with the RNA primers. okay? But before we get into that, I want to mention that if you have access to the electronic text, there are some videos in the electronic text starting with section 10.3 that show a lot of this, and they're actually pretty solid animations. I can't figure out how to download them or get them. If that's something you don't have access to but would like to, perhaps we can talk in the office hours and see if anybody figures out how to share them, or I could definitely share my screen during a Zoom call and we could watch those and make sure that everybody follows along, okay? All right, so let's talk a bit about the removal of these RNA primers and replacement. What are we going to replace it with? So here's where we left off. Down at the bottom, we have our original strand, and we've got these newly synthesized strands, and they're broken into fragments, some of them, and they have primers at the ends. So let's just pretend like we're finished. Top there is the original template, split it apart, made new strands. You can see that we've got 2 Okazaki fragments in the new strand, the upper new strand, and we've got a continuous long fragment on the bottom new strand, but there are 3 primers in there, okay? And we don't want our DNA slap full of RNA, okay? You cannot have RNA in our DNA, so we've got to get those out of there. So the first thing that happens is those primers get cut out, okay? And so DNA polymerase, there are different polymerases, it depends on the organism, but polymerases will go in and cut out that primer, okay? So you can see we've got a gap, primer A, B, and C. So primer A, super easy to fill in. Why is primer A easy to fill in with DNA if you're a DNA polymerase? What was the problem that necessitated primer A be created in the first place? The reason that we had to put a primer in the position A is that there was no DNA to the right of it with a free 3' end that the first nucleotide could be attached to. But if you look at it now, there is that second new strand to the right ends in a 3' end. And so DNA polymerase can go in and start adding nucleotides to the end of that rightmost fragment, okay, right up to the butt end of that leftmost fragment, okay? So DNA polymerase does that, gets rid of those primers, and then fills in the gaps with DNA because it doesn't have to be primer anymore. There are 3' ends that can be filled in. All right, so the gap A is filled, all right? And recognize that DNA polymerase is adding nucleotides to the end of what I will call arrow B, the top right one, to the arrowhead there. So growing it from right to left. And DNA polymerase will take that right up to the very end of the beginning, I guess I would say, of fragment A, the upper left one. But there's going to be that last little gap, because all DNA polymerase does is it adds a new nucleotide to the hanging 3' end to our right. What it's not going to do when it puts in that last nucleotide is also connect the 3' end of that new nucleotide to the 5' end that's already there on that leftmost fragment. And so that little tiny gap has to be spanned, that connection, that phosphodiester bond, has to be made using another enzyme called ligase. Ligase is, you can think of it as a molecular glue. Okay, so gap A gets filled through the action of DNA polymerase removing the primer and ligase gluing together that last little phosphodiester bond. But we still have C and B unfilled. Why could we fill A but not B? Because there's nothing to the right of B. The reason we needed a primer in the first place was there was no 3' end over to the right side, and there still isn't. But we went ahead and got rid of the primer. That seems premature. So let's think about what this means for the very tips of our chromosomes, the ends, okay? What it would mean if we just left it like this on the bottom is that the very tips of our chromosomes are single-stranded, okay? And let me tell you right now that single-stranded nucleic acids are not stable. They get chewed up, beat up, broken down, broken off, okay? And so what we would end up with if we left it this way is that every time the DNA was replicated, we'd leave a little single-stranded end hanging off, and then that end would get broken off. And so over your lifetime, how many times do you think you've replicated your DNA? If your chromosome got shorter every single time, you'd start losing important stuff, okay? And that's where we start talking about telomeres. Telomeres are the very tips of your chromosomes, and they're indicated here in red in this illustration. And telomeres are really interesting. They protect chromosomes. There are no genes there that we know of. There's not a lot going on. They protect your chromosomes from fusion or being degraded, from the ends being broken down. I think of them like the bumpers on your car, okay? They're there to protect. And there's some really interesting health implications for the health of your telomeres. There are some interesting genetic conditions. If you may have seen some TLC special on TV, or you might be aware of a genetic disorder called progeria, okay? Like geriatric means old, progeria. It means early onset aging. And individuals born with progeria, go ahead and Google it. You will see that these people, by the time they're 10 or 12 years old, they look like a 90-year-old person, okay? And progeriacs have prematurely shortened telomeres. They don't have all their telomeres. And this was taken as evidence, direct evidence, for the influence of telomeres on aging. So despite the fact that I'm about to talk about a way that our telomeres maintain themselves and retain their buffering capabilities, over time, it probably doesn't work 100%, and it does seem that the shortening of our telomeres throughout our lifetime contributes in part to our aging. So the reason that you can outrun your parents and do things that they can't is because you're young, but what makes you young? It's your ability to rejuvenate and regenerate. And that shortening of telomeres that comes with time contributes to the aging of the body. So, unsurprisingly, there's a new enzyme we're going to learn about, and this enzyme is called telomerase. And what it does is it goes to what is, in all of us, a very G-rich region, okay? You'll notice that this single-stranded bit hanging off that was left when we removed that primer has a lot of G's in it, okay? And the telomerase has a little bit of RNA in it that is complementary to bits of this G-rich tail. You can see here that it has a little stretch AAC that are complementary to the very tip of this single-stranded bit of DNA, okay? And when the telomerase binds here, complementary to the DNA, this allows us to come in and fill in this gap, okay? Because what telomerase has done, two things happen, okay? Telomerase allowed there to be complementary nucleotides, these 6 new pink GGG-TG, to be put in there. And look at what it ends with. It ends with what telomerase had attached to in the first place, TTG. Okay, so telomerase adds to this long pink overhanging end. So that's not really what we were looking for. We weren't looking to waste a lot of energy making this single-stranded end longer. We want to make it double-stranded. But by making this single-stranded end longer, it lengthens the gap, okay, that you can see in the bottom. And this process is repeated over and over and over again making that gap longer and longer and longer. And once it's long enough to go in and put a primer and synthesize a new chunk of blue DNA. Okay? So if you make that single-stranded pink bit really long, it allows you to go in and add a double-stranded component of DNA. And if in the end you end up losing a little bit of the tip that's single-stranded once the telomerase goes away, let's say you fill in exactly what is indicated by this parenthetical gap at the bottom, and then telomerase goes away. How many pink nucleotides are left hanging? 4, 8, 10? Yeah, 10. 10 of those nucleotides are left hanging. Let's say they get chewed off or broken off, like I said is quite likely. Doesn't matter, okay? We've actually extended the length of our chromosome. It's a few nucleotides longer than it was in the past, okay? Remember, it used to end in that black TTG. Now we've added 2 nucleotides, okay? So telomerase, once it's released, primase comes in, puts in this new primer, and we synthesize DNA, so you can see the new DNA that's been put in there. This gap is sealed. You'll remember what seals that gap. That's the ligase. Okay, so DNA polymerase fills in the gap, ligase seals the gap, okay? And then instead of us having left all of those black letters just hanging and getting broken off, once we get rid of this new primer, the CCAAC that are in orange, what are we leaving hanging? Just those 5 nucleotides at the end, okay? And sure, they get broken off. Who cares? Our chromosome's longer. Our telomeres are longer. We've got a better bumper on the end of our chromosome, okay? So in this way, we can deal with the primers that are introduced in both the leading and the lagging strand, the internal primers and gaps that are created, and then also the gaps at the very ends. So understanding the role of DNA ligase, DNA polymerase, telomerase are all quite important in understanding how DNA is replicated. Now, the last thing I want to bring to your attention is that replication occurs at multiple points simultaneously. Our chromosomes are enormous, and if we simply started at one end and worked our way to the other, it would take a long, long time. And so what happens is replication bubbles form throughout our DNA, and replication forks push in both directions, okay? So in this long strand of DNA, what's happening at all of these replication bubbles is that replication is proceeding both to the left and the right. And if you look at that very top red arrow, you will see that 2 different replication bubbles are about to meet, okay? And so once they intersect one another, the replication is done, okay? Those gaps will get cleared of primer RNA by DNA polymerase and gaps filled in, and they are going to keep pushing in opposite directions, but they're done filling in the gap in between. So you can imagine even if you have a really long chromosome number 1, if replication starts in hundreds or thousands of points along its length, DNA replication can proceed very quickly. Even in suboptimal conditions in my lab, in a machine, in a tube, I can get DNA synthesis at rates approaching 2,000 nucleotides per minute. So that's how fast polymerases can work. They add new nucleotides at the rate of about 2,000 per minute. Super fast. Okay, so that's it for Chapter 10 and our review of how DNA is synthesized.